It sure is really awkward to just stand here and wait for a minute. My name is Didem Sarikaya. I'm your next speaker. Now, today I'll be talking about work that was conducted in Cassandra Exelmore's lab, which is my PhD lab, with the following collaborators. And I'll be talking to you about reproductive capacity evolution in Hawaiian Drosophila. The major goal of Evo-Devo is to look at changes at the cellular and genetic level to understand morphological diversity that we see in nature. And one of the recent movements in evolution in Evo-Devo is to also look at the ecology of the animals and think about what are the ecological factors that are driving morphological evolution in an approach called Eco-Evo-Devo. And today I'll be telling you about my PhD work, taking an Eco-Evo-Devo approach in understanding evolution of Drosophila ovaries. Above, you're seeing pictures of ovaries from different species of Drosophila, and I hope you can appreciate that there's quite a variation in size. The functional unit of the ovary is the ovariole for insects, and this is true for all insects. And this in particular is what a Drosophila ovariole looks like. At the very anterior tip, there's a germline stem cell that will divide every now and again to give rise to an egg, and the oocyte will mature as it goes through development, and what you see here in white elongated structures are actually the matured eggs that are being waited to be oviposited. When you're going in, that ovariole number is species-specific and genetically determined, and that it positively correlates with fecundity. So you can think about them as egg-producing factories, and so species that have more of them have the capacity to produce more offspring. And you can also think about the fact that this could really lead to different life history approaches to the animal if it has very little reproductive capacity versus if it can produce 100 or 200 eggs a day. Within the genus Drosophila, the number that we currently know ranges from 2 to 100. And I wanted to understand how does this trait evolve? And specifically, I focused on a group where most of this range comes from, which is Hawaiian Drosophila, and the data is highlighted in pink. Hawaiian Drosophila is a canonical island radiation. 1 to 2 ancestral populations are thought to have given rise to over 1,000 species with incredible morphological and behavioral diversity. These species are categorized into monophyletic groups within Hawaiian Drosophila in these 5 groups here. And one unique feature of these groups is their reproductive ecology. So Hawaiian Drosophilas have very specific egg-laying substrates specific to native Hawaiian plants and parts of the native Hawaiian plants. So AMC subgroup species are leaf breeders. They primarily lay eggs on the decaying leaf matter at the forest bed. Polyacylus species are fungus breeders. Modified mouthpart species, depending on the species, are leaf, bark, fruit, or sap flux breeders. In this case, what I mean by bark are either decaying stems on a tree or a fallen tree that's decaying. So as the tree decays, it turns into pulpy mush, and the larvae will feed through that. Picturings are primarily bark breeders with some sap flux breeders. Scaptomides are primarily generalists, denoted by this purple circle, with some really cool exceptions. They have leaf breeders, flower breeders, and spider egg breeders. And what the spider egg breeders do is they specialize on these spiders in Hawaii that have these stringy egg sacs, and the female will go in and oviposit onto the egg sac, and the larvae will feed through the eggs as it develops. A study in the 70s documented ovariole number from various Hawaiian Drosophila species. What you're seeing here are averages across the number of species that he documented. In parentheses are the number of species that was documented in that study. And one of the things that the author, Mike Camposelos, noted was this difference in ovariole number depending on the egg-laying substrate. So he noted that the bark breeders had a much higher ovariole number compared to the other species, and flower breeder that he had data from had really low ovariole number, the lowest possible number, which is 2. And he postulated that perhaps larval ecology has an effect on ovariole number evolution. Perhaps the ephemerality and the stability of the food source or the abundance may be driving this diversity. However, at the time, the statistical tools were not available to test this, and also the data set is a little sparse in some of these key groups like Scaptomides and modified mouthpart. So building on this study, I wanted to investigate how ovariole number evolves in Hawaiian Drosophila. I addressed 2 questions. First, what are the ecological and evolutionary parameters influencing ovariole number evolution? And second, what are the developmental mechanisms that give rise to this diversity? I conducted fieldwork in Kauai, Maui, and Hawaii Islands with the below collaborators. And we brought the flies back to the lab, tried to get them to lay eggs, and I also collected adult traits like ovarial number, body size, and egg size. And before I talk about the data, I do briefly want to mention that ovarial number is genetically determined in the melanogaster subgroup. This is very well documented. And in my data set, which is limited, I do believe that this trait is also genetically regulated in Hawaiian Drosophila. This is data comparing F1 offspring in blue that were raised eating the same food and the same in the same temperature in the same environment, so like a common garden experiment, comparing ovarial number for the F1 offspring and their moms that were caught in the wild, and I don't see a significant difference between them. This is a data set that we collected. We expanded significantly for Streptomyza, modified mouthpart, added more species of AMC, and also some new picture-linked species. And we conducted some computational analyses to test this. The analyses that you're going to see in the next few slides were done by Sam Church and Cassandra's lab. And the trees that we used were generated by Laura Lab and Marcino using data that was published from Patrick O'Grady's lab. There we go. So first we looked at the correlation between body size and ovarial number. We conducted a PGLS analysis over 100 posterior distribution beast trees. And we found a positive significant correlation, suggesting that larger flies tend to have more ovarials, and that allometric growth of the ovary may influence ovarial number. I do want to point out that this does not explain all of the variation. So you have species like the Anthropoceras flies here, which are really big but have low ovarial number. But it does explain part of the trait. We next looked at ovarial number and egg volume, so how large the eggs are, and found a significant negative correlation, suggesting that flies with higher reproductive capacity make smaller eggs, and there may be a potential trade-off there. And lastly, we looked at the correlation between ecology and ovarial number, and for this we did an Ornstein-Uhlenbeck analysis, testing for changes in evolutionary optima of the trait. We tested 3 models, OU2, where bark is coded to have its own evolutionary optima compared to all the other food sources. OU3, where we coded bark, flowers and spider eggs, and all the other as having their own evolutionary optima. And we based this on bark being potentially the most stable food source and flower and spider eggs being more ephemeral. And OU7, where each of these food sources had their own evolutionary optima. We conducted this analysis again over 100 beast trees, and we were able to reject the 2 null hypotheses, so Brownian motion for random evolution and OU1 for there being 1 evolutionary optima, and both of these were rejected in our analysis. And we received support for OU3 and OU7, suggesting that reproductive ecology does influence ovarial number evolution, but we may want to do some more extensive studies on adding more species to test which of these, or test other models to further understand how this trait's evolving. Okay. So next I looked at the development of the ovary. So early on in my PhD, I identified this particular group of cells called terminal filament cells, which I'll be referring to as TFCs, as being really important for determining ovarial number. So here you're seeing in red, they're labeled with engrailed antibody staining. They emerge early in development and intercalate to form these really nice stacks that are outlined in that white dashed line. When you look at the adult structure, that corresponds to the very tip of the ovarial structure called terminal filaments, or TFs. And what happens during development is that these cells surrounding the TFs move down into the bottom of the screen. Oops, well, I went ahead of myself. To engulf all the other cells. The TFs serve as a beginning point for an ovarial to form, and they do the punchline away, but my work and works from other labs have shown support that ovarial number is determined by the number of terminal filaments that form during development. And I compared development of some melanogaster subgroup species and found 2 developmental mechanisms that can change the number of these terminal filaments. First is through sorting. So if you imagine we start with the same number of cells, but one species puts fewer cells per stack, that would result in more terminal filaments. So you're dividing the same number by a lower value, so your output would be higher. And through changes in cell number. So instead of changing the number of cells per stack, we have the same numbers of cells per stack, but we start off with more cells, and that leads to a higher number of terminal filaments. So I tested whether sorting or terminal filament cell number influences ovarial number evolution in Hawaiian Drosophila. This is my larval data set. It is considerably smaller because it is quite difficult to get them to lay eggs, but I was able to collect data representing a good number of the egg-laying substrates. The terminal filament number shown here correlates almost one-to-one with the adult ovarial number, as we would expect. First, I looked at the parameters for sorting and found that the cell number per stack ranged from about 6 to 12. But when we looked at the correlation between cell number per stack and terminal filament number, we did not observe a significant correlation, suggesting that sorting is not really a process that's relevant for the evolution of this trait in Hawaiian Drosophila. Lastly, I looked at total cell number, which ranged from 30 to 300, and we found a significant positive correlation with cell number and terminal filament number, suggesting that total TFC number influences the number of these terminal filaments, thereby determining ovarial number. So to summarize, I started with this question of understanding the diversity of these ovaries that we find in Hawaiian Drosophila. And the first conclusion that I was able to draw is that reproductive ecology is influencing ovarial number evolution. So visually, you can see this is an ovary from a flower breeder, and this is an ovary from bark breeders, and these are all corresponding. And you can visually see quite a big difference as you go through the different egg-laying substrates. Second, I identified that these changes are happening through cell numbers. So bark breeders have a tendency to have more terminal filament cells as opposed to flower breeders and the other intermediate ecologies. And that this change may be partly mediated by allometric growth. So we're still testing some of these hypotheses, but there's some indication that depending on the species group, ovarial number may be coupled to body size. So there may be a pressure to just make bigger flies and they will have bigger ovaries. But this doesn't explain all of the variation in the trait, so that's why there's a dashed line here. And just to appreciate the diversity in size that you see, these are flies hanging out on a sponge, and they're quite different in size, and it's really remarkable, the evolution that they've undergone. With that, I would like to thank my PhD lab, specifically Cassandra Extor, who's been extremely supportive throughout all these endeavors of doing field work, and my amazing collaborators. You can find me at BioDids and Twitter. I'm currently in the Begun Lab at UC Davis. My collaborator Carl has a website, hawaiianinsects.com, if you're interested, and Sam is giving a poster. And with that, if you guys have any questions, I would